Today, Dave McMullen, co-chair emeritus of the Toronto and District Multiple Myeloma Support Group, is, as you know, a man of all talents, knowledgeable, dedicated, hard worker, and kind. Dave chaired this support group for over 10 years, founded the Patient Advisory Council and chaired it, and currently is chairing the Multidisciplinary Committee of Myeloma Canada. And he's on the CMRG committee, and I cannot tell you all. I probably he will not have time for his presentation. As I said, he also told me to make it short. Today, you can also read in our newsletter about the recent award of many that Dave received. Dave is a recipient of this year's Exceptional Leadership in Patient Involvement in Cancer Research Award. So relevant for today because CAR-T, as you know, the CAR-T is still in trials. Dave's presentation today is about CAR-T cell therapy, a patient perspective. I can't say enough about Dave's friendship, advocacy to our cause, and positive attitude. When I visited Dave at PMH during his CAR-T admission, I was pumped by his positive attitude that I'm sure you will hear today. He encouraged me when I came to visit him. So go figure. But you all know Dave for so long and so many years. So without further ado, I would like to invite Dave to present to us his patient perspective to CAR-T cell therapy. Thank you, Dave.
Thank you, Renat, for those very, very kind words. So, about the CAR-T experience. Again, I was so humbly grateful that I had the opportunity. I just happened to be relapsing and met the quite stringent inclusion criteria for the trial. But I didn't do it alone. This is a picture of Erika and me. Please don't tell anybody, but this picture was taken 12 years ago when Dave had more hair, but Erika hasn't changed. But my dear wife Erika has been with me every step of the way, since I was diagnosed 12 years ago, and of course, very much so in the CAR-T process. So, what is CAR-T? I'm sure you've all heard of it. There we go. Put that back. Yeah. It's an abbreviation for what's called a chimeric antigen receptor. T cells. T cells are a part of your immune system, and they are modified, re-engineered to become what's called these chimeric antigen receptors. And what it is antigen is a type of protein that's on the surface of myeloma cells, in this particular case. Chimeric means that they are being changed so they have gene features from not just me, but also others. The receptors are what grab onto features on the myeloma cell. What happens? Your own T cells are harvested using a pheresis process similar to what you have for stem cell transplant, and sent off to a special manufactory to be genetically re-engineered to have these CARs. And they enable the T cells to be directed to a target, such as what's called BCMA, and I'll explain that a little bit later, on the surface of the myeloma cells. What are called lentiviral vectors are used. And that's about the extent of my knowledge in virology, but in a nutshell, lentiviral means slow incubation period, viral, and vectors, it means you use these viruses to make changes to the genes. So what happens then after the virus does its thing? on some of these T cells that are collected. The manufactured cells are then expanded. That's the term that's used. But basically it means mass produced, so that you have many. And then they're sent back to the hospital to be reinfused in the patient. And the CAR T cells then latch on to the myeloma cells and attack and destroy them. There are side effects which do occur. They're usually fairly well managed. It's still an area of research. The whole process of CAR T cell therapy and even managing the side effects, the medical community is still learning as we go. Here's a diagram that kind of explains the process. What is CAR T? Down here, this represents a myeloma cell, and it has features on the surface. Proteins called antigens. It has quite a variety of types of antigens. The one that applied to the trial I was on is called BCMA, or B-cell maturation antigen. And here's a T cell, but it's modified with this CAR. And what this CAR does is it grabs onto the target, the BCMA antigen, so that the T cell can then work at destroying the myeloma cell. And again, T cells are a normal part of the immune system. I think I've explained that these chimeric antigen receptors are modified specifically to enable the T cells to recognize that target. As I mentioned, there are other targets on the myeloma cells. CAR T is used for some types of lymphoma and leukemia. There, it's a different target on the cell, so the CAR T cells are modified to suit the particular target in mind. So this describes ideal targets at the top of the page here. You don't have to read it all, but for B-cell lymphomas and leukemia, there's a feature on the surface of the cells called CD19, and that is the target for the CAR T cells. For myeloma, BCMA, the B-cell maturation antigen, is a good target. There are others, but so far the BCMA is the one that has been most frequently used. And it targets the myeloma cells in particular without targeting and damaging other cells in the body. So please don't try to read all the small print, but this describes in a summary of the entire process. For me, there was a lot of screening tests because it was part of a clinical trial. But aside from that, there still might be some testing. But the first part of the process is called leukapheresis. Pheresis, again, is the procedure that is used to collect the cells. And by the way, if anybody has any questions en route, please feel free to ask, and we'll have time for questions afterwards. Leuka is a name for white blood cells. So they're harvested and they're sent off to the manufacturing facility. In my case, it was a big facility, a big lab in California. The T cells are extracted from the white blood cells, and then the reprogramming takes place so that the T cells have these receptors, the CAR receptors, as I mentioned, that enable it to attack the cancer cells. And then this expansion takes place in the lab where these are mass produced. Then they're sent back to the hospital. And for me, it was Princess Margaret in Toronto. And before they are reinfused into the body, there is a process called lymphodepletion. That means depleting the lymphocytes, which are part of the immune system, by chemo, so that the body can take on these modified T cells without rejecting them. And it's a little bit like the melphalan that you get prior to a stem cell transplant. It's not as strong or not as onerous. But there are, 2 chemo drugs that are used, quite consistently, everywhere in the world, I believe. Cyclophosphamide and another drug called fludarabine that are used, and it can cause some side effects, nausea and the like. It wasn't too bad for me. And then the T cells, after 3 days, basically, of waiting, T cells, the modified T cells are reinfused into the body. So that's the summary of the CAR T process. And our history, when I say our, my dear wife Erika was as much a part of the journey as I was. I was diagnosed in 2012. I've had 2 stem cell transplants and numerous other treatments, including Revlimid, daratumumab, pomalidomide, carfilzomib, cyclophosphamide. I had Revlimid for induction for the transplant, first transplant. And I've taken part previously in 2 clinical trials with bispecific antibodies. I think you've probably heard of this type of treatment that is still really quite new. It's still very much in the research world. That there are a couple now that have been approved for use by the Food and Drug Administration in the United States, and some other authorities like Health Canada approves them for use. It doesn't mean they're funded. That's a whole additional process. But anyway, I was fortunate enough to take part in trials with 2 of these bispecific antibodies. One was called cevostamab, and that worked quite well for me for about a month. And then it stopped working. It works quite well with most people. I was just in that 35% category for whom it didn't work very well. And the same thing with the next one that I took. It's called talquetamab. And for that, I was in the 30% there where it didn't work at all. But I don't regret at all taking part in those trials. You don't know till you try. There are some people, likely some people in this room, who have had the bispecific antibody treatment, and it's worked very well. So great treatments, it just didn't work very well for me. The CAR T process has been around now for a few years, as I mentioned, for some types of lymphoma and leukemia. It's been approved by Health Canada and funded by the provinces. Newer is the T cells for myeloma, and there are actually 2 commercial products that are used significantly, when I say that, significantly in the United States, not so much in other countries because they're quite expensive. But these 2 products here, one called Ida-cel, another one called Silda-cel. I've got the brand names there as well, and the manufacturers. These have both been approved by Health Canada. The Ida-cel, that was not recommended for funding by the provinces by an agency called CADA that makes recommendations to the provinces for that, in part because the remission time from trials was not very long, like about a year as a median remission time. This one here, the Silda-cel, has shown a much better remission time. You might remember Dr. Chen in April. She talked about this and said, so far it appears like a median of approaching 3 years, which is really quite encouraging, especially considering that people who take these treatments probably have already had quite a large number of previous lines of therapy. In other words, they've had therapy, relapse quite a number of times. And oftentimes for patients who have had a lot of previous therapies, new therapies don't work as well, on average. But keep in mind, everybody is an individual. None of us are medians. So this one here, cilta-cel, it was recommended for reimbursement by the provinces, and there's negotiations underway now with the provinces as far as what the price is going to be. We're hopeful it's going to be approved sometime before long, but being realistic, I'm not going to hold my breath. And one reason I say that is that if you look at European countries, like some of the wealthier European countries, there's only one country so far that has approved funding, public funding, for the cilta-cel, and that's Germany. And Germany tends to fund almost everything. They've got a somewhat different, what's called a health technology assessment system than most other countries. And the reason for that is that it's so expensive. So, in my layperson's opinion, it'll come at some point, but it might take quite a while to go through the pricing negotiation. And there are many other companies and research organizations in many countries developing CAR T-cell therapies. Clearly in the United States, some in Western Europe, Israel. However, there's a new kid on the block, relatively, called CARSgen Therapeutics from China, which has developed a new CAR T-cell product for myeloma. It doesn't have it does have a name. It's called Zevercel, or CT053. I mentioned ide-cel, cilta-cel. Well, this is Zevercel. It's had phase 1 and phase 2 clinical trials in China, but now, in the last year and a bit, they want to do trials. These are like early phase style trials at a few centers in North America. And our context here is different. Like in China, they just don't have the same access to all the new treatments that we have here. So the Chinese have developed CAR T. When I first heard of this CARSgen company, I was wondering, coming from China, is it really, does it have credibility? Well, yes, the Chinese are world leaders in CAR T-cell technology, and they're developing a number of other types of CAR T processes. But because, again, the context is different here, like people who take part in who have access to CAR T currently, like even in the United States, have had more lines of previous therapy. CARSgen Therapeutics wanted to do some trials here. How long is it going to last? How long is the remission going to be? We don't know yet. The data's not in. So I guess I'm part of that data that will be used to determine what a median remission time is. I mentioned why I was selected. Have to be more or less in the right place at the right time. You have to have a certain number of prior treatments. You have to be relapsing, and still have good blood analyses. Things like your hemoglobin platelets, neutrophils, creatinine, and a variety of other parameters. Be in pretty good health. Again, the screening requirements are really quite stringent. So I was offered, and it took me about 2 seconds to think about it before I said yes. I had already done some research, to know what it entails. So I'm just going to go through the overall process. In summary, it's reviewing and signing the informed consent form, initial screening, which is quite extensive for any trial of this nature, a washout period, which means that you can't take any anti-myeloma drugs for a certain period before this leukapheresis process, and likewise before the reinfusion of the CAR T cells. The manufacturing process done way off-site in another country. Bridging therapy, most patients, because they are relapsing, they need something to contain the myeloma so it doesn't go way out of control. The lymphodepletion, as I mentioned, that's the dose of chemo prior to having the CAR T cells infused and the infusion, and quite a long recovery in either in the hospital or some patients now are doing it outpatient, where they have to be close to the hospital for about a month, and then afterwards. So the informed consent form, this is something that is done for every clinical trial. It's a requirement where the patient and the family are given the form and take it home, review it, ask any questions. It describes in language that lay people can generally understand what the trial is about, what its purposes are, what the potential side effects are. It's a little bit like the insert you get with the box of Revlimid. It gives you a long list of potential hazards, and I think they even, say death, is not a very likely situation, but they have to put it down, right, so that you're aware of what the possible risks and hazards are. The various tests that you have to undergo, like bone marrow biopsies, I've had a number of them so far. So here is, that's the first page of a 28-page document. Please don't try to read it, but that's kind of what it looks like. The most useful part of that document is what's called a sequence of events table. So this is it here. And it describes some of the various activities, some of the screening tests and other tests, and at various times during the trial process, like screening, even before you are admitted to take part in the trial, a baseline test. They want to know what your status is, what the myeloma status is, and other parameters before you have the CAR T, the lymphodepletion, and what's called day 0 is the day that you receive the cells. It's kind of like with the stem cell transplant. Day 0 is the day you receive your stem cells back again. And then it describes various procedures that you have for weeks, months, and up to 15 years afterwards. So they really want to keep an eye on you for a long time. And again, lots of activities. It really is quite a busy time. We made many visits to the cancer center for these various studies. PET scans. I didn't have any plasmacytomas. That's myeloma growing outside of the bones. Bone marrows, yes, had a number of those. Quality of life questionnaires. That's something that is part of the trial. How are you feeling? And there's actually 45 questions that they ask. And it's not onerous to complete, but that's good. They want to know, basically, how's your quality of life and how is it being impacted by this treatment. Pulmonary function tests, pregnancy if you're a woman, lots. Sometimes I had 16 vials of blood that were gone, and most of them would go to the sponsor, go to Car's Gym. For their information. I mean, they're paying for the trial, so they want to learn as much as possible about what this treatment does, to what they call the kinetics of the blood. Do a mental assessment. How sharp are you or not? Because the treatment can affect your cognitive abilities. Yeah, vital. I mean, often, I've lost count of the number of times I've had my blood pressure and temperature measured, but that's a very frequent procedure. So again, Screening, as I mentioned, you need to have for this trial, you need to have prior therapies, including a proteasome inhibitor, like Velcade or Ixazomib, carfilzomib, or an IMID, like Revlimid or pomalidomide, and daratumumab. And again, that's one thing that's different. In China, they don't have access to things like daratumumab, so that's why they want to find out how it's going to work in North American patients who are a little further in their journey and have had some of these treatments. You need to be refractory to the last treatment. In other words, it's no longer working and you're relapsing, and the disease is measurable usually by blood analysis. You can't have any prior BCMA treatment without a response. I think if you had prior BCMA, bispecific antibody, and you had a good response, you might be eligible, but I don't know that for sure. And you have to be reasonably healthy, and again, many blood tests that you need to pass. So I passed them all. Sometimes, is a little bit iffy. Is my creatinine going to be good enough, or my platelets high enough? Yes, they were. And lots of physical examination, 24-hour urine, of course, electrocardiogram, echocardiogram, and I mentioned these before. Washout, which means no anti-myeloma treatment. They want to make sure that when they do the trial, that the effectiveness is not in any way going to be influenced by other treatments that I might be having. Not even dexamethasone, which is kind of nice being off dex for a while. So, 2 weeks prior to leukapheresis, is there any possibility that the dexamethasone or daratumumab or carfilzomib or whatever you're on, it could anyway impact your T cells? They wanted to eliminate that possibility as much as possible. So I had the leukapheresis October 25th last year, and it's much like collecting stem cells prior to a transplant. And those of you who have had a stem cell transplant will recognize this scene. This is the pheresis machine, which is like a big centrifuge where they collect, in my case, it was all the white blood cells. There's a happy customer there. One thing that's different from the stem cell transplant, as you know, those who have done it, you're given in the transplant process some drugs to mobilize the stem cells for a period of 5 to 10 days in advance. Things like Grastafil or Neupogen, or in some cases cyclophosphamide. That's not needed for this process. You just go there. I think they run, I believe it's 2 complete cycles of all your blood through the centrifuge machine and take what they collect. Other than that, it's very similar. And then the harvested T cells, all the white blood cells, were sent to the manufacturing facility in California. As I mentioned, the T cells were separated. And the manufacturing period typically takes 4 to 6 weeks. Sometimes it takes a little longer, which can create a little bit of anxiety among some people. With some of the newer approaches that are in research, they're looking at reducing that manufacturing time frame. I heard of a Chinese product called FastCar, C-A-R, right? Where it's reputed to be 48 hours. Now, how well it works, I don't know. But anyway, there are studies looking at reducing the manufacturing time. Because in the meantime, myeloma patients, they're relapsing, and we don't want the myeloma to get out of control before the reinfusion. So there's very stringent quality control in the manufacturing process and transportation. My cells were closely monitored. First, they're frozen down to minus 150 degrees C, and very closely monitored on their way to California, at the manufacturing facility, and the way back. But there's always a question: could this manufacturing process be botched? Yes, sometimes it happens. Not very often, but I've heard of people. I'm on a Facebook page with other CAR T patients in the United States and elsewhere. And yes, sometimes it does botch, and they have to, reharvest again. Are my CAR T cells going to make it back to me coming across the border? In my case, they were held up in airport customs for a little while. But the team at Princess Margaret were very expeditious in overcoming that obstacle. So bridging therapy, as I mentioned, most patients need it, and there's a few selected treatments that can be used. It's not a very long period, but it can be important for some patients. While I was waiting for the return of the cells, I was very cautious. I didn't go to any support group meetings, or I had a number of other engagements planned. I canceled them all, unless it was something I could do, virtually by Zoom. And in my case, I was relapsing quite significantly. My free light change, the kappa went up to 5,400, and hemoglobin was dropping. That's a sign that the myeloma is becoming more active. And I had 3 new lesions seen by the PET. So I asked my doctor, what if it doesn't work? He said, well, it's going to work, Dave. But if it doesn't, there's selinexor out there, might have to use D-paste to mow the grass, so to speak. In other words, to debulk the tumor, as they say. In other words, cut back the myeloma before using selinexor, which is a slower acting drug. And there are other therapies that are used, some of them still in the research world, that might be available by clinical trials. A few that drug companies are offering on a compassionate basis that aren't funded yet. But something, analytical nerd like me, I ask these questions. Anyway, it all worked out very well, fortunately. So baseline tests, this is to see what your condition is before the treatment. And again, quite a few tests as part of the trial, physical, mental assessment, lots of blood analysis, 24-hour urine, bone marrow biopsy. I'm getting quite used to these bone marrow biopsies. Brain MRI, because these T cells cross the blood-brain barrier. So you need a brain MRI before and afterwards to see if there's any changes in the brain. PET scan, electrocardiogram, and these quality of life questionnaires. Do you feel better or do you feel worse? Again, with the lymphodepletion, this again is the chemo that's required to knock down your lymphocytes so that you can receive the T cells. So there's a washout period of 3 weeks required prior to that. I had a PICC line placed. I'm just going to get a little drink here, which was very handy. I found the PICC line much more convenient than having the central line that goes into your neck and chest. But it's a very handy way to do blood draws and the like, and to have any type of transfusions. And then a fairly large dose of chemo, as I mentioned, and everybody's different. Some people feel quite unwell with this. For me, it wasn't too bad. I had a bit of mild nausea curtailed with ondansetron, which is an anti-nausea drug. That it tends to, for me, it tends to get me bunged up a little bit, but it wasn't a major problem. So the lymphodepletion process is 3 days, and 3 days of cyclophosphamide. Like I think you're all probably familiar with that. You've taken that as part of the Cybard-D in your induction treatment. And another chemo drug called fludarabine, which is, to my knowledge, not used elsewhere in myeloma treatment, but it's quite a standard drug that is used for lymphodepletion for all types of CAR T. It was done outpatient, starting day -5. So for me, that was December 2nd, a year ago today. So I'd go to the clinic and then go home each day until it would be December 5th last year. I was admitted into the hospital. They want to make sure that you're settled and everything looks good before doing the infusion, which took place at 11 o'clock in the morning on December 6th. No, I didn't notice anything. No side effects. There's the package that's my CAR T cells came in from the manufacturing facility in California. And there's approximately 180 million cells there in the package. And mixed with 20 milliliters of fluid, DMSO, which is the same product that's used in doing stem cell transplant. And as I mentioned, these are cryopreserved, in other words, at a very low temperature, minus 150 degrees C, and warmed prior to the infusion. The infusion process didn't take very long, about 10, 15 minutes. And as I mentioned, I didn't notice anything that day. Lots of monitoring. They watched me like a hawk, and I must say, most kindly. I'm just so grateful at the level of care and concern and kindness that they gave me in the hospital. So no side effects on day 0, +1, +2, or +3. But lots of assessments, including cognitive ability. Is Dave losing his marbles or not? Well, I didn't, although Erika would be the best person to answer that question. Lots of prophylactic drugs taken, during this period, and basically, some of these carried on for various periods, like in some cases for the first month, and in some of them I'm still on. But antibiotics, I did get a fever, so they gave me some intravenous antibiotics. I got a fever on day 4. Now, antifungal medications, there's a risk of having fungal infections. That was part of the regimen. Antiviral, it's like acyclovir. It's called valacyclovir. It's stronger than the regular acyclovir that some of you, I'm sure, are taking. There's a type of pneumonia called pneumocystis jirovecii pneumonia, which CAR T patients can be susceptible to. I was given an antibiotic, not initially. Initially, because the blood counts come down to almost 0, but as soon as the blood counts started to recover, I was given a special antibiotic called Septra, and I'm still on it today, 3 times a week. As I mentioned, the T cells can go through the blood-brain barrier. There's a risk of having seizures. You're given an anti-seizure medication for the first 30 days. It's called Keppra. Some of you might have, if you're at all familiar with epilepsy, that's not an uncommon drug for that. I need to be on a blood thinner because some of the previous medications that I had, and I've had clots. So it was a 16-day period recovery in the hospital. Initially, they were planning not to have me in the hospital. A fair number of patients now are doing it, the whole process, outpatient. They have to be close to the hospital. They might be renting accommodation close to Princess Margaret or close to wherever the center is that's doing the CAR T. And you come there every day. And there are a lot of pluses. You sleep in your own bed. There are bugs in the hospital, right? But for me, because this was my 8th line of therapy, and I was relapsing quite significantly, that they thought, well, better keep Dave as inpatient. They treated me very well. I felt a little odd, though, because in the hospital there are sick people, and I really felt generally okay. So then on day +4, I developed a fever, and that is a symptom from what's called cytokine release syndrome. Actually, I was glad to see that, because then I knew that the CAR T was doing something. Just as an example, for the previous bispecific antibodies, I didn't have any cytokine release syndrome, which tends to happen frequently with those therapies as well. So cytokine release syndrome is common. With the two myeloma CAR T cell therapies that I mentioned earlier, with the ide-cel, that typically happens a day or two after infusion. With the cilta-cel, it's about 7 days. So for me, it was 4.5 days, which is kind of in between. So I had a fever of 38.9, which is not really high, but it's showed something was happening. I generally felt okay, maybe a little bit lackluster. I had no nausea. Some people do get significant nausea, but it wasn't particularly impactful for me. One thing that did happen, my blood pressure got a little bit low. So one night I went into the bathroom and I was about to sit down in the toilet, and I feel quite embarrassed about this. But anyway, I lost my balance and fell forward and braced myself with my hands. But my head grazed the floor. And as you know, the bathrooms in hospitals, they have, the floor has an abrasive surface, so that it's not slippery. So anyway, I grazed my forehead, and again, kind of embarrassed. I didn't think I should be, falling forward. So I didn't say anything, but it just so happened that a short time later, the nurse came in and noticed that I had, quite a red mark and swelling in my forehead. So I had to confess what happened. Anyway, before long, I was whisked down to have a CT scan of my head, because at this point my platelets were really quite low. The chemotherapy and especially the chemotherapy and also the CAR T process, it does reduce your blood counts, including platelets. So again, they're very cautious, erring on the safer side. Went down for a CT scan at 3:30 in the morning, and as it turns out, everything was okay. There's no internal bleeding or damage. But it's an example of how they go the extra mile just to make sure that I'm safe. I had this mark for quite some time. Sometimes I jokingly described it as my Gorbachev mark. Apologies to anybody here who might be Russian. The cytokine release syndrome lasted for 4 days. And what it is, cytokines are small proteins in the blood that direct the immune system of what to do. I mean, I'm sure they do more than that. And what happens with the CAR T? These CAR T cells, the 180 million, which actually grows to a much larger number once they're inside the body. They tend to release these cytokines, and they can be inflammatory. And there's one there, it's called interleukin 6 that can become quite prolific. So there's a drug here, a monoclonal antibody called tocilizumab, or sometimes abbreviated as TOCI, that is commonly administered for patients who do have significant cytokine release syndrome. So I had that. Initially, talked to the doctor, should we do it, should we not? But then Dr. Crum, who is a doctor who is very experienced with CAR T therapy at Princess Margaret, mostly lymphoma and leukemia, he said, no, we should do it. So, and it reduced my fever, almost immediately. I still had a bit of fever for another couple of days. So, to my knowledge, I had no neurological toxicity, no impact on cognition or anything else. Some people, it can be significant. I've heard of some people that had to be intubated because they just lost, needed that help in breathing. Or some people, again, would lose their cognitive ability. But I don't think, Erika, I've done that, but you're a better judge again. I don't know. Please comment.
Well, I'll just mention one thing that was noticed that during his fever, the writing became a little bit smaller. So there was a neurological change, if you want to use that as a indicator. But fevers do affect everybody. So it was just a small thing, but it was noticeable to me.
Yeah. Well, my thought is that I just had lower energy, so I didn't write as big. But anyway, Erika's probably a better judge. So again, please don't try to read all this, but normally the symptoms from CRS, as it's called, are fairly mild, like mild flu-like symptoms like nausea, fatigue, headaches, chills, and fever. But it can result in these more serious symptoms, like low blood pressure. I had mildly low blood pressure, but it can be worse. You can have a fast heart rate, capillary leakage, other cardiac conditions, cardiac arrest, arrhythmias, and something here that I don't know what it is, but it doesn't sound very pleasant. So that's why they watch me like a hawk. Not just because it's on a trial. I mean, they want to, that's part of the mandate on the trial, is to learn everything. But anybody who has CAR T afterwards is going to be watched closely by their medical team. I mentioned about the neurological toxicity. There's a condition here called ICANS, or immune effector cell neurotoxicity syndrome. That terminology is used anywhere in the world, as far as I know, where they do CAR T and in many cases the bispecific antibody therapies as well. It can be life-threatening. You can have seizures, and go as coma, as I started to describe. You can have tremors, shakiness. I've got a friend in the United States who had Bell's palsy. Yeah, Bell's palsy. Thank you. Bell's palsy and other symptoms. So had to do a test frequently. Not hard, but with some of these things here, but I had to pass 10 out of 10. Otherwise, in the hospital, they would call the doctor, and when I went home, Erika had to administer this test. She was instructed, if Dave doesn't get 10 out of 10, call us right away. And there's not much in the way of treatment. High-dose steroids like Dex is one of them that might be used. So again, I was in the hospital for close monitoring, lots of blood tests, vital signs. Because I had an elevated temperature, they looked very closely for any types of infections and gave me lots of supportive medications that I'll come to. But generally, I felt pretty well with a reasonable appetite. The neutrophils, just like people who've done a stem cell transplant, they hover slightly above zero, and they didn't want to give me any Neupogen or Grastafil, thinking that could cause cytokine release. At least not at that point. After a few days, they did give me some Grastafil, and just like with some of you have experienced with the stem cell transplant, I did need a blood transfusion and platelets. So there were a number of things that went a little bit awry, not very much. And because of that, they kept me in the hospital longer than what normally might be the case. So they wanted to keep a close eye. Some of these were actually, these little things going awry were a result of adverse effects from the medications. I had mildly elevated liver enzymes, so they did an ultrasound of the liver, and it seems to be okay. One drug gave me a slow heart rate, So they took me off of that, put me on another drug. And some of them might affect my neutrophils as well. When it came close to being discharged from being an inpatient, Erika and I had to attend CAR-T school so that we could manage, or I could say Erika could manage me at home. And there are quite a number of protocols that we need to do. So I was an outpatient at the hospital for 4 weeks, which means roughly twice a week I'd go into the hospital. For those who had transplant outpatient, it's the auto transplant day unit. You know, the same wonderful nurses that treated you treated me. And there were times when I needed some transfusions, the GCSF, that's like the Neupogen or the Gracefil, sometimes other meds. And the days that we didn't go in to the auto transplant day unit, we got a phone call every day from a nurse, you know, asking all kind of a standard script of questions, but, you know, a very nice way, you know, how are you doing? How's your blood pressure? You know, how's your temperature? How's your appetite? How's your bowel movements? And you know, just making sure we're okay. And as I mentioned, it's somewhat like a stem cell transplant, not as onerous. Like for me, my appetite was not significantly affected. But the blood counts for me were slower in recovery than was the case for the stem cell transplant. And that's, you know, might be because I've had, you know, so many other lines of therapy. But the immunoglobulins, like in my case, you know, my type of myeloma is IgA. It was and still is a very low value, now less than the normal range of IgA. The IgG is also low, and because of that, I take immunoglobulin replacement therapy so that the IgG is in a good range. The kappa and lambdas are very low, like below normal range for both of them. Well, again, at home, we had to do you know, vital signs, take blood pressure, and a few other things that I'll show you every day for 30 days. No driving. Wasn't allowed to drive a car for 8 weeks. So my dear wife, Erika, took me everywhere. Took me to the cancer center and anywhere else we had to go, which is, you know, we're not used to that, right? Having to depend upon somebody else for driving and for somebody else having to do all the driving. Advised to stay in isolation for 3 months. In other words, not go out to parties or gatherings like this because I'm susceptible to infections until the immune system recovers. Generally felt pretty well. Sometimes a bit tired. You know, the hemoglobin was low, and that's one reason why I would feel a little tired. But there was an amazing response. You know, I mentioned that my kappa was, you know, 5,400. It was probably higher. And within a very short time after the CAR-T infusion, it went down to less than 2. There's a sensitive measurement of whether or not you have myeloma. It's called immunofixation. That's more sensitive than the SPEP test that you get to monitor for you know, myeloma proteins. And it was immunofixation negative, which is very good. They did the bone marrow biopsy and they couldn't find any myeloma cells in the marrow. They, to my knowledge, didn't do a more sensitive study called minimal residual disease. The sponsor might have done that, but hasn't shared the results. But I'm going in week after next for another bone marrow biopsy, and they say they're going to do, you know, the MRD test. It's called minimal residual disease, and ideally, if it's negative, MRD negative, then that's great. But if not, you know, I've still had an excellent response. You know, and I'm just so humbly grateful. And that the last time we had the blood draw was almost 2 months ago. That's 10 months after I had the CAR-T therapy, and everything was still very good then. I don't know how long it's going to last. As I mentioned, I'm kind of a guinea pig. The data isn't in yet as far as how well it's going to work in the North American context. Every day is a blessing. So we had to keep this daily record. So this is the one for January 1st of last year. It was day plus 26. And as you can see here, 4 times a day, take the temperature, the oxygen saturation levels. They give you a little device that you clip onto your finger, and it measures how much oxygen you have in your blood relative if you are fully saturated. Heart rate, sitting and standing at various times. Blood pressure, again, sitting and standing. In my case, my blood pressure is normally quite low. Roughly, how much you drink, bowel movements, and the nurses ask you about that. So it's important to maintain these records. Here's the test I got twice a day from Erika. These questions aren't very hard, like what year is it, what month is it? Didn't ask what day it is. That would be too hard a question. What city are we in? What hospital are we going to? Name 3 objects, like a pen, a cup, and do something that's challenging, like close your eyes and stick out your tongue. Not very challenging, but if you've got this ICANS situation, you might not be able to do it. Count backwards from 100 by 10s, 190, 80, 70, etc., back down to 0. And handwriting. Is your handwriting, is it legible? Has it changed much? So again, I had to pass this test 10 out of 10 every time. Otherwise, Erika was instructed, call, and they probably asked Dave to go to the cancer center right away. Or at least, they would, grill me with questions, very politely, of course. So there, Erika talked about the handwriting. You're supposed to choose a sentence. I picked one that's quite simple, easy to remember. And as you can see, there wasn't a whole lot of difference there. But if sometimes patients could have tremors, or if the handwriting gets small, or if they forget about what they're supposed to write, then that's a warning sign that something could be happening upstairs, because of the CAR T treatment. So currently it's 12 months afterwards. Generally I feel great. My hemoglobin is still lowish, like last time I was 103. And that means that my aerobic capacity is lower than normal. I can still, go out and walk. I can jog a little bit, and do some aerobic. We went hiking in the mountains, and I can do that. But I have to go slower than normal and breathe more deeply than normal. So it's not really a significant limitation. My doctor tells me it could take, a year and a half, 2 years for the hemoglobin to start recovering. And that's, again, just kind of collateral damage from the CAR T, but it's something that's certainly very tolerable. Sometimes a little tired, and actually I wrote this a little bit earlier. Disclosure, I've had a couple of practice runs for this presentation. I did it with the Holt Appeal group in June and the in-person and the Edmonton group by Zoom in October. So really, I rarely feel tired now. Some patients do feel tired for quite some time. And it's great to be off the anti-myeloma drugs, especially things like Dex. Dex, for me, it really caused the muscles to atrophy. So I've been going to the gym and regaining some of the muscle strength. Vaccinations, it's much like after a stem cell transplant, although in my case, because it's part of a trial, they watch more closely. Things like the DTaP, the tetanus percussive, diphtheria, Hib, that's a type of pneumonia, meningitis. They test your antibodies for hepatitis. In my case, they were okay, so I didn't need hepatitis. Pneumonia, a series of 4 shots for pneumonia. And as soon as I was eligible, like 3 months afterwards, I got the COVID vaccination, and they start you right from, step 1, as if you've never had a COVID vaccine before. Shingrix, they recommend that, and the flu shot, which we waited until the fall. But anyway, I've done all of those, and that's part of the protocol. And as I mentioned, every day is a blessing. So what about CAR T in general? What's the future? Well, in my layperson's opinion, there's tremendous hope. There's a lot of research taking place for CAR T and related therapies. I'm involved in a couple of projects. There's one that's called TAC-T, which is T-cell antigen coupler. It's like CAR T. I'm involved in another one called gamma delta T-cell, which is not CAR, but it's another T-cell therapy, similar. There's in Ottawa, they're looking at CAR-NK. NK cells are another part of the immune system, another immune system cell. One thing that's happening now in Canada, actually, it's been happening for a few years now in the lymphoma world. There's an organization called BioCanRx, which has been working with, in Ottawa, that's been working with other researchers in Vancouver and Victoria and Montreal, and they developed a lymphoma treatment called CLIC-01. That's Canadian lymphocyte immunotherapy for cancer. Canadian lead. Must be a few of the CAR T cells in my brain. Canadian lead immunotherapy for cancer, which is an amazing. It's amazing that these folks have done this. Where in Ottawa they developed the special viruses, and then another facility in Vancouver developed a process. That there is a system that you could probably put on this table that does the manufacturing process. It's a system bought from Germany. So there have been, I don't know how many, but last time I heard that there were more than 30 lymphoma patients that had this treatment. For some of them, it was literally life-saving. So that type of future, it's called point of care. Now, in this case, it wasn't point of care because the virus came from Ottawa. The manufacturing was done in Victoria, BC. But the concept of every major cancer center having a device that you can put on a bench like this to do the CAR T cell manufacturing. I mean, that is really encouraging. It would increase the supply and lower cost. So that's something that I know Myeloma Canada is really interested in being a catalyst in doing more research into this type of therapy. And there's quite a bit going on in Canada and other countries in the world. In Spain, they've had quite a bit of success with their own approach to point-of-care CAR T-cell manufacturing. So right now, as I mentioned, the remission period for the ida-cel so far is only about a year or a little bit. Cilta-cel, on median. Some patients do a lot longer. Some it doesn't work at all. With the Cilta-cel, its median is about 3 years now. It'd be nice if it's longer. And the same thing, by the way, applies to lymphoma CAR Ts. The majority, more than half of people who get lymphoma CAR Ts do relapse after a while. it often sometimes works better with young people, like children or adolescents. So in general, the CAR T process is wonderful, but it's still being developed. As I mentioned here, it's not a panacea, not yet, but it holds encouraging promise for myeloma and for other cancers as well. So the hope is for increased durability. In other words, it lasts longer. The safety concerns, as I mentioned, the cytokine release syndrome, and the other, the ICANS, the effect on the brain. Hopefully that will be reduced. And some of these other methods that I mentioned, the gamma delta, that holds the hope of having reduced of these side effects. And availability. There has been limits to the ability of the large pharmaceutical companies to manufacture a lot of CAR T cells. I think that's being reduced now, but the fact that there are very few countries in the world that fund it. In the United States, if you got lots of money or a very good insurance program, you can get it. But in most other countries, really, it's not available through governments, not yet. So I mentioned the cost is very high. There's limits in the manufacturing, and having these bench style point of care manufacturing facilities, that should increase the supply. There's also the concern about the supportive care. Having, especially if patients are inpatient, taking up beds and taking up, the time of highly qualified healthcare professional, doctors and nurses and others. Our healthcare system is already overstrained. So this is a concern. So part of the research is looking at ways of reducing the burden on our healthcare systems, like doing it outpatient more and learning more about how to handle the possible risks and side effects. I mentioned here CARTITUDE-5, the trial, that there is a trial that's underway now. I'm not sure if it's still recruiting. For newly diagnosed, they're looking at how well this might work for newly diagnosed patients. Again, I'm not totally up to date there, but if anybody is newly diagnosed and interested, it's something to discuss with your doctor. So that's really all I had, and I would be delighted to take any questions. Not too long, because there's lots of food there we want to eat. But anyway, whatever. If anybody has any questions, I'm kidding about the food. I'm not going to prevent you from asking questions. The food will be there.
Dave, one very quick question.
Yeah.
I agree with the food. On the day that they put your T cells back in, did you have any side effects? Like sometimes I had my last stem cell transplant, my blood pressure got so low that I required oxygen. So was there anything like that?
No, not for me. Now, for some patients, I've heard that, they did notice something, but the volume of the fluid that is infused, like when you had your stem cell transplant infusion, you probably had a significant volume of fluid. Here, it's only 20 milliliters, not very much. I didn't notice anything. But, again, every patient is unique. Yes, John?
You've had a lot of different therapies.
Yeah.
Was the CAR T transplant one of the roughest ones?
No, the roughest one would be the stem cell transplants.
That would be even rougher.
Yes, yeah. It's not as, for me, and for most patients who have done the CAR T, they'll say that the CAR T is not as onerous as the stem cell transplant. There are some exceptions, but generally that's the opinion of patients who have done both.
Also, because of the stringent requirements to go into a therapy like this, age limit would be an age exclusion?
To my knowledge, no. Thank you. Thank you for mentioning that. Yeah, good, thank you. John just asked, is there any age limit for the CAR T? To my knowledge, no. They'd be looking Now, every center is different. There might be some centers where there are, but generally speaking, as far as I know, they look at your health status. They have the various screening tests and the like, baseline tests, but if you're healthy, to my knowledge, there's not an arbitrary limit. Like if you're over 75, sorry, no, it's to my knowledge, it's looking at your health status. Yes, Patricia.
I was wondering if a person came out of remission after having CAR T therapy, would there be any possibility of having a second line of CAR T therapy, or because of expense, would that be excluded as an option?
Good question. I wondered that myself, right? Because probably, I'll come out of remission at some point. And right now, unfortunately, it's really not available or funded in Canada. But there are patients that I'm aware of who have done 2 CAR T cell therapies. I asked my doctor about that, and from a medical point of view, one of the criteria is how long did the first one last? If it's like a stem cell transplant. If it didn't last very long, then they wouldn't do a second. I'm sorry, Patricia asked, could a person do a second CAR T if the first one, if you relapsed after the first one? So I would say it's I would say so. Like I've heard of American patients who have really good insurance programs, or lots of money, doing that, but it's not a, in fact, doing the first CAR T is not a common practice. Yes, Patrick.
Can I flip Patricia's question around and kind of allude this to the talk, but to what extent can a CAR T or some of the other things that are being developed be considered a cure?
Okay, you ask, can CAR T or some of the other therapies be considered a cure? My own observation, I don't think our myeloma doctors, they're trying to be realistic, and they don't like using the cure word, just not to get expectations too high. My own sense as a layperson, we're not there yet. We might be sometime. However, if you do a CAR T or any other treatment, I've got a good friend who's been on thalidomide for 20 years. Well, she might eventually die of something else. Was she cured? Okay, you can say that. But we're not there yet with, even with lymphoma, you hear of especially young people getting CAR T and 10 years later, they're survived. I think sometimes the cure word is used, but I don't think you'll hear that from medical professionals. Not yet, but stay tuned. Yes, Engen.
Glad to see you responding to CAR T, and you're in good remission so far. I'm just wondering, are you on any maintenance therapy?
Okay. The question is, am I on any maintenance therapy? No, it's not part of the protocol for this trial. But there are other trials where maintenance therapy of various types are used. Even things, I think, like, maybe Revlimid, although most people who have taken a CAR T trial have already, had Revlimid and used it up, so to speak. But yes, there are various maintenance therapies that are used for some trials, but it's not, to my knowledge, it's not a common practice in the clinic. Yes, Rosa.
How many patients so far, Princess Margaret, have received this CAR T therapy?
Oh, not very many. There are just a few slots available. Princess Margaret is the only center in Canada that's, as I mentioned, there are just a few centers that CarsGen wanted to have this trial, and it's very expensive to run clinical trials of that nature. So there aren't very many slots available. That's what it's called, a slot. You're asking how many people at Princess Margaret have had this therapy. I don't know, but it's not a very large number.
If it is not funded by the government,
I'm sorry?
if it is not funded by the government, what may be the cost?
The cost? Well, you're asking what does CAR T cost? I really don't know. Like the sticker price, I've heard of a figure of $475 US. I don't know if that's true or not. And generally speaking, the actual price might be negotiated downwards by the insurance company. In Canada, if anything is publicly funded, the pricing is always negotiated downwards from the sticker price. But it's very expensive. That's one thing that makes these point-of-care therapies attractive, is that the manufacturing process can be a lot less. Now, there's still a cost on top of that for all the supportive care needs, and the cost of doing the pheresis, the cost of doing the lymphodepletion. So that is added on to the cost of the manufactured product. Yes, ma'am.
Dave, thanks so much for your presentation. It's been very interesting and informative. My question is, and I know you don't know, because you already said you don't know, but do you have any sense of when CAR T may be approved for publicly funded approval in Ontario? I know you said you thought it would be a ways away, but is that 1 year or 5 years? Do you have
Well, that's.
sense at all of where we are in the process?
Yeah, well, I understand that there's you're asking when is CAR T likely going to be funded here in Ontario or in Canada? For myeloma, yes. And again, I don't know. I understand that, for the cilta-cel that I mentioned, there's a pan-Canadian organization called the Pan-Canadian Pharmaceutical Alliance, which does negotiations on behalf of the provinces, and they've recently picked up the file and negotiations are starting. So, fingers crossed, it could be fairly soon, like within the next year. It may not be, and the reason why I say that, I look elsewhere in the world that have publicly funded healthcare systems, like most of the Western European countries, they're not there yet. A lot depends upon the pricing. Now, as a patient, I would argue that a 3-year remission period is saving the cost of other expensive drugs during that time. And there's some patients who will have a remission for a much longer period. Okay, well, that's, you do the math, and that's an important part of the equation. So, certainly, that would be part of the negotiation process. So, I really don't know. I'm optimistic, I'm hopeful, but, at the same time, I want to be realistic too.
So do we have, I'm sure they're doing some pharmacoeconomic studies and analysis. Do we have any information with other blood cancers from the time that they were recommended for funding to the time that they were actually approved?
Well, the best, I guess, comparison would be for lymphoma. I don't know what the timeline was there. Time between it was approved by Health Canada and reimbursed by provincial governments, some provincial governments. I think the math was a little more favorable with lymphoma because of the the median duration of remission. But to be honest, I don't know what that time frame is.
Thank you. Thanks again for your presentation.
Yeah. Well, thank you. Yes, please. Yep.
You may have mentioned this, and I'm not really sure, I apologize, but how often do you go in for checkups now at this point?
Yeah. The question was how often to go in for checkups. Well, initially, as I mentioned, it was once every twice a week actually, and then it became once a month. Then in early fall, it's gone to once every 2 months. I understand, if things look good, it might go to once every 3 months, sometime next year. But, every time, they take, the 16 vials of drugs, 16 vials of blood, 24-hour urine, and as I mentioned, I'm going to have a bone marrow biopsy and a PET scan, week after next. And that's all required as part of the trial.
You don't get a bone marrow biopsy every time.
No.
Okay.
No. But yeah, it's. I showed you the sequence of events, and the bone marrow biopsies, PET scans, and the like are there, along with the frequency of the visits. So, yeah, next year it should be once every 3 months. Any other questions or thoughts? Yes, Ellen.
Regarding the cost of the drug, it's not only just the cost of the drug. One of Dr. Rodriguez's presentation is that also trying to find the drug itself. She gave a ballpark figure of $500,000.
Right.
And then the supportive care actually costs about $500,000. So you're talking about $1 million for this one treatment. And she said the difficulty is finding an extra $500,000 of supportive care money for each patient. That's where the block might be. Other than the approval of the drugs for coming from.
Right. So your comment was that in addition to the cost of the manufactured cells, there's the cost of the supportive care, which can also be very high. Yes, that is a concern. But as I mentioned, through research and also clinical practice, looking at ways of reducing that cost so that the entire package can become affordable. Like, you mentioned a figure of $1 million, $1 million Canadian dollars. That's a lot. Like, you can do a lot with $1 million in having life-saving therapies and elsewhere in the healthcare system. So clearly there's a need to bring the cost down, not just the cost of the product, but the cost of the supportive care. Well, thank you. Thank you very much. And